The research paper introduces a novel deep learning model, White Matter Geometry-guided Diffusion, WMG-Diff, to address the issue of missing tissue microstructure data in diffusion magnetic resonance imaging, dMRI, tractography fiber clusters. This model aims to tackle challenges such as interindividual anatomical variability and the quality of neuroimaging scan data. The WMG-Diff model consists of three key components: a deep score-based guided diffusion model for tissue microstructure imputation, a white matter atlas geometric relationship guided denoising function to guide the reverse denoising process at the subject-specific level, and comprehensive training and evaluation on a large dataset with 9,342 subjects. The authors demonstrate that their proposed WMG-Diff model outperforms state-of-the-art methods in both tissue microstructure imputation and a downstream non-imaging phenotype prediction task. This work has significant implications for improving the accuracy of parcellation in white matter tractography, which is crucial for disease prediction, anatomical tract segmentation, surgical brain mapping, and non-imaging phenotype classifications. Brain parcellation, specifically white matter fiber clustering, is a widely used compact representation for statistical and machine learning analyses of white matter. However, fiber clustering can be inconsistent across datasets due to variability in scan quality, subject motion, interindividual brain anatomical variability, and brain development and aging, leading to missing microstructure data values. Recent methods, such as predictive mean matching and graph neural networks, have been proposed for imputing missing functional connectomes and morphometry values, but there is a lack of prior work on imputation approaches dedicated to fiber cluster or diffusion MRI microstructure data. This work investigates the imputation of fractional anisotropy values from an anatomically curated, atlas-based fiber cluster parcellation applied to a large, harmonized diffusion MRI dataset. Score-based diffusion models, which have been successful in various deep generative modeling tasks, are employed for imputation. The score-based diffusion model uses two Markov chains, where the forward process introduces noise into the data, and the reverse diffusion denoising process learns to reconstruct the data to impute missing values. The conditional score-based diffusion model, an extension of the score-based diffusion model, provides informative guidance toward the target and is effective at learning the conditional probability distribution of the observable values. This work extends the score-based diffusion model by leveraging information about white matter fiber cluster geometry to define a guided denoising function. A novel guided denoising function is proposed, which replaces the random data reverse diffusion process with a geometry-guided reverse diffusion process. This approach utilizes prior knowledge of fiber cluster geometry to provide accurate imputation of missing microstructure data values. The use of diffusion models in diffusion MRI microstructure analysis is a novel contribution, and the proposed guided denoising function marks an advance in leveraging diffusion models to analyze microstructural properties. The White Matter Geometry-guided Diffusion model, WMG-Diff, leverages anatomical relationships between white matter fiber clusters to guide the diffusion process for imputing missing diffusion MRI microstructure data. By focusing on fiber clusters similar to those missing in each subject, WMG-Diff is hypothesized to impute more accurate tissue microstructure values. This approach has three main contributions. Firstly, it introduces a deep generative approach using a conditional score-based diffusion model to impute missing microstructure values, a novel application in the field of dMRI microstructure imputation. Secondly, it utilizes anatomical information of white matter geometry to create conditional masks that aid the reverse diffusion generation process. Specifically, a guided denoising function informed by the geometric relationships between WM tractography fiber clusters is proposed, utilizing information from anatomically similar fiber clusters. The method is evaluated on a downstream testbed sex prediction task, demonstrating that WMG-Diff outperforms popular state-of-the-art imputation methods on a harmonized, large-scale dataset. The framework used to impute missing fractional anisotropy values guided by geometric fiber cluster relationships is presented in Figure 1. The study utilizes a large dataset of fiber cluster microstructure measures, created by generating whole brain tractography for each subject, parcellating subjects' WM fiber clusters, and extracting their quantitative FA measures. The geometric relationship between each pair of fiber clusters across the whole brain is calculated as a guided reference to the denoising function of WMG-Diff. The model is evaluated on the large-scale, multi-site adolescent brain cognitive development dataset, which includes 9,342 young children aged between 9 and 11 years. To standardize the dMRI data across 21 different acquisition sites, the data is harmonized to remove scanner-specific biases while preserving intersubject biological variability. The FC bundle parcellation employed is consistent across subjects, demonstrated by its application across the human lifespan, across disease states, and across different acquisitions. The paper introduces a novel pipeline, WMG-Diff, for processing diffusion magnetic resonance imaging, dMRI, data to predict biological sex. This approach utilizes white matter geometric distance and fiber clustering to enhance the accuracy of sex prediction models. A dataset of 9,342 subjects, comprising 4,879 males and 4,463 females, was used in this study, divided into training and testing sets for five-fold model evaluation. Tractography and fiber clustering were performed using a two-tensor unscented Kalman filter method, allowing anatomically sensitive estimation of brain connections. The resulting tractography was then parcellated into 953 fiber clusters using the org anatomically curated tractography White Matter Atlas. Microstructure measures, including fractional anisotropy, FA, were computed for each streamlined point from the multitensor model employed to perform tractography. The white matter cluster features were calculated as the mean FA value of all points within the cluster and input to the proposed framework. The geometric relationship between the fiber clusters was quantified using a white matter tractography distance, a common strategy widely used to cluster fibers. This distance describes a cluster's geometric relationship with the other clusters, as illustrated in Figure 2, which shows 3 sample clusters from the org atlas. The proposed WMG-Diff pipeline imputes missing FA data for input to a downstream sex prediction testbed task, as shown in Figure 1. This approach aims to improve the accuracy of sex prediction models by leveraging the geometric and microstructural properties of white matter fiber clusters. The authors quantify geometric similarity in white matter tractography anatomy by calculating the minimum average direct flip pairwise streamline distance between each pair of fiber clusters in the org atlas. This distance metric is illustrated in Figure 2, which shows the pairwise distances between 3 example fiber clusters and the resulting matrix of distances between all 953 white matter fiber clusters. To address missing fractional anisotropy, FA, data in tractography analysis, the authors propose a geometry-guided score-based diffusion model. This generative model learns the conditional probability distribution of observable values in the dataset, providing accurate imputation of missing FA data. The model consists of convolutional layers, a diffusion embedding module, transformer encoders, and residual blocks. The training dataset is composed of 3 parts: actual missing FA data, non-missing training data randomly divided into guided observable values and imputation targets, and synthetic missing data with known ground truth values for self-supervised training. The model's residual block is conditioned with geometric guidance, training the denoising process through a continuous iterative diffusion process. This estimates the true conditional data distribution of the imputation targets given the guided observable values distribution. By leveraging prior knowledge about white matter structure, the model imputes missing FA values while considering subject-specific tissue microstructure distribution, enhancing the learning process about missing data. Researchers have developed a novel method, WMG-Diff, to impute missing fiber data in white matter diffusion MRI, leveraging geometric relationships between fiber clusters to guide the training process. This approach addresses the issue of missing data due to anatomical or scan protocol variability. The guided training process involves selecting observable values and imputation targets based on geometric distances calculated from a white matter atlas. For each missing FA data value, K-guided observable values are chosen, representing 80% of the non-missing training data. This procedure utilizes information about WM anatomy and the distribution of missing fiber clusters to direct the training process effectively. The WMG-Diff architecture involves inputting actual missing data and non-missing observable data, which are then split into observable values and imputation targets using cluster geometric relationships. At each time step, the imputation target undergoes a forward process to add noise, becoming a fully noisy target. Subsequently, the reverse diffuse process utilizes guided observable data to denoise the noisy target and minimize the difference between the noise and the output. By leveraging geometric relationships within WM anatomy, WMG-Diff offers a promising solution for enhancing the quality of MRI scans and improving our understanding of brain structure and function. This innovative approach aims to improve the accuracy and reliability of diffusion MRI data analysis by addressing the problem of missing fiber data. The application of a white matter geometry-guided diffusion model for imputation is explored in the context of a downstream sex prediction testbed task. This model utilizes microstructure values generated from the WMG-Diff model as input for a logistic regression model to predict non-imaging phenotypes through neuroimaging features. The prediction task is defined as a binary problem, serving as an additional metric to understand the real-world applicability of the generated data. The model's implementation details include the number of diffusion layers, attention heads, and other parameters. It is trained and evaluated with a specific batch size and number of epochs, using an NVIDIA RTX A5000 and PyTorch version 1.13. The performance of the model is quantified using the root mean squared error, RMSE, for the missing FA data imputation task and the accuracy, ACC, for the sex prediction testbed task. The proposed WMG-Diff model is compared with common imputation methods in neuroimaging analysis, such as replacing missing data with the mean, median, or zero, as well as two open-source state-of-the-art imputation methods: generative adversarial imputation nets, GAIN, and multivariate imputation by chained equations, MICE. In the realm of neuroimaging data, missing values can significantly hinder analysis and interpretation. To address this, various imputation methods have been developed, including the proposed WMG-Diff model. This framework leverages the conditional score-based generative adversary network (GAN) architecture to impute missing data. A comprehensive comparison of imputation methods reveals that WMG-Diff outperforms state-of-the-art approaches and conventional neuroimaging data imputation practices. As evidenced by the lowest root mean squared error (RMSE) value in Table 1, WMG-Diff demonstrates superior performance in imputing missing data. An ablation study further examines the efficacy of the geometry-guided denoising function, showcasing that incorporating geometric relationships into the denoising process yields better outcomes compared to randomly masking data. The results of a downstream testbed sex prediction on imputed missing data, presented in Table 2, highlight the proposed method's exceptional accuracy, surpassing that of all other imputation methods. The WMG-Diff model's strength lies in its ability to capture complex patterns and relationships within neuroimaging data, enabling accurate imputation of missing values. By leveraging the conditional score-based GAN framework, WMG-Diff effectively balances data fidelity and diversity, resulting in superior performance. The geometry-guided denoising function plays a crucial role in this process, as it refines the imputation process by incorporating spatial relationships between neighboring voxels. The White Matter Geometry-guided Diffusion (WMG-Diff) model is a novel conditional score-based diffusion model that utilizes the reverse diffusion process to impute missing FA values in brain imaging data. This model leverages prior knowledge about white matter structure and geometric relationships between white matter fiber clusters to enhance learning during the reverse diffusion process. Evaluations on the ABCD dataset demonstrated that WMG-Diff outperformed widely applied state-of-the-art methods. Moreover, when the imputed data was used for a non-imaging phenotype prediction task, WMG-Diff showed superior performance over widely applied imputation methods for the sex prediction testbed task. This suggests that the proposed WMG-Diff model can generate accurate tissue microstructure data, which further benefits downstream tractography-related prediction tasks. The study adheres to ethical standards, using public ABCD imaging data with approval granted by the BWHIRB. The research is supported by various National Institutes of Health grants and other international funding bodies. The Adolescent Brain Cognitive Development (ABCD) study is a comprehensive imaging data collection across 21 sites, aiming to provide insights into brain development and cognitive functions. This section highlights the importance of harmonized diffusion MRI data and white matter measures from this study. The authors have developed methodologies such as Tract-GeoNet, a geometric deep learning framework that analyzes tract microstructure to predict language assessment performance. Deep fiber clustering is another approach introduced, an anatomically informed unsupervised deep learning method for fast and effective white matter parcellation. Additionally, the authors discuss Tract Graph Former, a hybrid graph CNN transformer network that uses anatomical information for classification from diffusion MRI tractography. This methodology is compared with other machine learning regression algorithms and sample sizes in predicting individualized behavioral outcomes with functional connectivity features. This section also reviews various other studies related to the ABCD study, including the use of quick bundles for tractography simplification, and the application of deep learning approaches to model and predict age and sex in healthy subjects using brain white matter features. Overall, this section emphasizes the significance of the ABCD study in providing a large dataset for researchers to explore and understand the development of the adolescent brain. It also showcases the development of novel methodologies that leverage this data to advance our knowledge of brain structure and function. In this research paper, we delve into the realm of advanced machine learning algorithms and their applications in various domains. The primary focus is on deep learning techniques, which have revolutionized the field with their ability to automatically learn complex patterns from data. The paper begins by introducing the concept of neural networks, a fundamental component of deep learning. It explains how these networks are structured, how they learn from data, and their role in tasks such as image recognition and speech synthesis. The authors then explore different types of neural networks, including feedforward, convolutional, and recurrent neural networks. Each type is explained in detail, along with its strengths and weaknesses, and examples of real-world applications. One of the key contributions of this paper is the development of a new algorithm for optimizing the training process of neural networks. This algorithm, called DeepOpt, significantly reduces the computational time and resources required for training large neural networks. The authors demonstrate the effectiveness of DeepOpt through a series of experiments on various datasets. Another significant aspect of the paper is its discussion on the challenges and limitations of deep learning. The authors highlight issues such as overfitting, vanishing gradients, and the need for large amounts of labeled data. They also discuss potential solutions to these problems, including regularization techniques and data augmentation methods. Finally, the paper concludes by discussing future directions in deep learning research. The authors predict that future advancements will involve the integration of deep learning with other AI technologies, such as reinforcement learning and symbolic reasoning. They also emphasize the importance of developing more interpretable and transparent deep learning models. Overall, this paper provides a comprehensive overview of deep learning techniques, their applications, and the challenges associated with them. It serves as a valuable resource for both beginners and experienced researchers in the field of machine learning. This section of the research paper offers a detailed examination of methodologies and applications in diffusion models, encompassing their role in missing data imputation, white matter tractography, and brain connectivity mapping. The authors provide an extensive list of diffusion models, such as the isotropic and anisotropic models, which are crucial for understanding the movement of water molecules within the brain. These models are essential for identifying white matter tracts and mapping brain connectivity, as they can reveal valuable information about the brain structure and function. The paper also delves into the application of diffusion models in missing data imputation, where they are used to estimate missing values in datasets. This is particularly important in neuroimaging studies, where missing data can significantly impact the accuracy of results. By utilizing diffusion models, researchers can fill in gaps in their data, leading to more reliable and accurate findings. Furthermore, the authors discuss the limitations and assumptions of these models, as well as potential future work in this area. They highlight the need for further research to improve the accuracy and efficiency of diffusion models, particularly in complex brain structures and dynamic processes. Overall, this section provides a comprehensive overview of the current state of diffusion models in neuroimaging and their diverse applications, underscoring their importance in advancing our understanding of the brain.